All right, so these are your notes for Chapter 5, Section 1 over the cell cycle. All right, so I'm recording these for you guys at State 1X. Hopefully you guys do a great job, break a leg. And if this is after the fact, congratulations, hopefully. But your objectives for this section are to describe the stages of the cell cycle, compare rates of division in different cell types, and to identify factors that limit cell size. So we'll go through a couple definitions here first. We've talked about a little bit of this in class already, but your cell cycle is that regular pattern of growth, DNA duplication, and cell division that occurs in eukaryotic cells. And we had four main stages that we had already gone over: gap 1, synthesis, gap 2, and mitosis. So when we were talking in class, trying to do our study guide already, gap 1 was when normal growth was happening. We also said that there should be normal functions going on in the cell at that time. So you might add that here if you want, but it's not a big deal. It'll be in there later too, so don't worry about it too much. Synthesis is when you're copying your DNA. Simply. Gap 2 is basically the same as gap 1. You're growing more. But there's also going to be these checks that we're adding now to talk about. And then finally, mitosis is that nuclear division itself. You have to divide the nucleus and the DNA in that nucleus so that that new cell would have its own nucleus with its own DNA to control everything that it does. So again, this stuff's just review, but there are a few more specific details that we're going to add for each stage along the way. So gap 1 on this slide, it does talk about the normal functions. Remember, if your cells aren't doing their normal functions, you wouldn't be alive. So that's actually one of the most important stages. This is a seemingly unimportant stage in terms of the cell cycle, because you're not dividing here, obviously, but this is what the cell should be doing most of the time. If you had a tissue, if you had 100 cells in a tissue, 99 out of 100 better be doing their normal functions, or that tissue isn't going to function the way it's supposed to. So it's really just a select few cells at any given time that are dividing. Most of them should be in gap 1. Right? So it says most of the cell's life is spent in this stage. That's going to be generally true. We do have to talk about a new thing, this G0 here, but that requires a little bit more depth. I'm going to draw a picture here to start to bring up that idea. So you will need this picture in your notes. I suck at drawing circles, so I'm going to cheat here, although I don't want it to be orange on the inside. So this is going to represent my cell cycle. So I'm going to start up here at the top, work my way around and back to the top. That's why it's a cycle. It goes around in a circle. And I'm going to cut this into chunks, right? This first chunk is going to be pretty big like this, and this is going to be my G1. Right? Again, most of the cell's life is spent in G1. It's a pretty big chunk in that cell cycle. I'm not really trying to draw this to scale at all. And in reality, we don't know exactly how long G1 is. I'll explain that a little bit more in a second. But most importantly, if it's in G1, it has potential to divide eventually. Right? That G0 is a whole different topic. But before we discuss that, we need to talk about how cells divide at different rates. Different kinds of cells divide at different rates. So you guys have experience with that. You know that, say you cut your finger, you get a paper cut, you get a small cut of some kind, that's going to heal fairly quickly, right? And the reason why that heals fairly quickly is because your skin cells replace themselves often, right? Your skin cells actually can manage to replace themselves, right, once every 2 weeks, and they have to, because you cause a lot of damage to your skin. You get cuts all the time, right? So your skin protects you from your environment. It's vulnerable. It needs to replace itself more often than a lot of other tissues. So if you get a cut of some kind, more likely than not, in 2 weeks, it's going to be healed. But some of you have probably experienced other kinds of damage. Maybe you've had a muscle tear or strain of some kind, and you know that that takes a little bit longer to get over. But that's because your muscle tissue doesn't really go through the cell cycle as fast as your skin does. Right? It only goes through the cycle about once every 6 weeks or so, which is why it takes 6 weeks to heal from that kind of injury. Right? And hopefully none of you are unfortunate enough, but maybe some of you have or know somebody who has been injured in an accident of some kind where maybe an internal organ was damaged, like your liver. And that's going to take even longer to fix because your liver cells only replace themselves every 6 months or so. So depending on the kind of cell, you might go through this cycle faster or slower. It just depends on what's necessary for that cell. But there are some cells that never divide again. Right? So when we're focusing on this G0 for a second, right, that's something we've talked about before in this class. Right? At a certain point, your development in your brain is going to quit. Right? Sometime between when you're 20 and 23, right, your brain is going to be fully developed and those cells will no longer divide, which brings up a problem. Right? If you have any damage to that tissue, it's going to be hard to repair it. So there's a reason why we prioritize that tissue, right? Why we want to make sure your brain's okay, right? In your spinal column, your spinal cord. Nerve tissues do not necessarily go through the cell cycle, right? They are still functioning tissues, hopefully, but if they get damaged, it's going to be hard for those things to be fixed, right? In fact, in most cases, it's impossible. So G0 is outside of the cell cycle, and it's entered into once that development quits in your nervous system. When your brain's done developing, all of those cells enter G0. They'll never divide again. So it's its own special thing that really only applies to a few select kind of cells. Most of your cells do divide and replace themselves, but not your brain tissue, not your nerve cells. So again, G0 is special. Right? Cells stop dividing at that point. Synthesis, right, was our next step. So I need to make a chunk in my picture for synthesis. It doesn't have to be a very big chunk. It doesn't take a long time for the cell's DNA to be copied. It certainly doesn't take the whole 2 weeks that your skin cells need to divide, right? That life cycle for them. But it doesn't take, it does take a little bit of time, but we'll talk more about how your DNA is copied later in other chapters. Right now, we're going to focus more on the other parts of this. So then my next chunk, G2. The only thing that's different there that we didn't talk about before are checks that are at the end of that stage. So in my picture, I'm going to add G2. Right? G2 is pretty big. That's more or less the same as G1 as far as I'm concerned. I think the book makes G1 way bigger than G2, but again, I'm not trying to make this to scale or anything. It's not supposed to be perfect. But G2 at the end of it is what we want to focus on. Right here at the end, I have some checks. So I like to think of that as just a stop sign, right? Before that cell divides, before it enters mitosis, it needs to make sure that a lot of things are in place and that that cell is ready to divide to create two viable cells, two cells that can survive after the fact. So right after this checkpoint, you would enter mitosis, but not if certain things haven't taken place yet. You have to make sure that that DNA has been copied accurately. you have to make sure that that new cell is going to have all the things that it needs. Because you might have the mitochondria to power one cell, but you might need to make some new ones to power your new cell. So lots of things have to take place before the cell can divide, and there's this important checkpoint at the end of G2 to make sure that those things happen. If they don't happen, those two new cells may or may not survive. So there's always going to be the stop sign right at the end. There should be, at least. If you don't have that, that's when the cell starts to have some problems. And we'll talk about those later. Mitosis itself, right, is divided into a couple parts, right? It's actually more than two parts. You have the mitosis, that's a division of cell nucleus and DNA. Technically, that mitosis has 4 parts that you may or may not remember. So I'm going to put those in there right now. We're not discussing them in this section, but there's a prophase, metaphase, anaphase, and telophase in mitosis. Right, so you may have talked about those things before in 8th grade when you were doing life science, but we're just going to go over those things in a little bit more detail now. But also, at the end of mitosis, there's that thing called cytokinesis. So, if you can't read how I wrote that there, it is on the next slide, right? Cytokinesis is the division of the rest of the stuff in the cell, right? The other contents, your organelles, the cytoplasm, that kind of stuff. So you have to divide the nucleus first, so you have 2 nuclei with complete DNA in each, and then you can divide the cell, then you have 2 new cells. So once you cross this line up here, I'm going from 1 cell to 2 cells, and those 2 cells start all over again in G1. Right? So they're going to start doing their normal functions, they're going to start growing. Eventually, you're going to copy that DNA. And then it's going to grow some more. You're going to stop to make sure it's ready, but that cell will enter mitosis one more time. So you keep going around and around the cycle to make more and more cells, following those steps. All right, so this question we answered in class, but I might have to go through it a little bit with you here. What could happen if the G2 checkpoint stopped working in cells, right? I just mentioned a moment ago about how those checks are necessary to make sure that that DNA has been copied accurately. Right, so I'm going to look for that. Right? DNA needs to be copied accurately, right? You need to make sure you have all the things that are necessary. There's lots of other stuff that needs to happen. Specifically, I mentioned the mitochondria, right? You need to have all the organelles that would be necessary for that new cell. So you might need to make some new mitochondria and whatever else for that new cell, right? So there's lots of checkpoints. Right? Most importantly is that DNA copied accurately or not. But another thing associated with this that we need to talk about, right, is that if those checkpoints are not working, that actually can lead to a disease, right? And it's a disease associated with the cell cycle because if cells divide uncontrollably, right, if they continue to divide and don't stop to do their normal functions, that's when you can get something called a tumor, and that would be associated with cancer. So cancer actually is a big connection to the cell cycle. We're talking about those G2 checkpoints. If they don't function the way that they're supposed to, you can end up with cancer. So I don't know how much you guys know about cancer. It kind of depends on the person. It wouldn't be a surprise to me if everyone in class knows someone who's been affected by cancer some way, somehow, right? All of you should, right? But cancer is a disease of the cell cycle. You have cells that are dividing uncontrollably, and it's a unique disease, right? It's hard to cure, right? The reason why we can't just cure cancer is because there's no two cancers that are alike, right? All we know for sure is that that cell is dividing uncontrollably. There's a lot of different damages that can happen to the DNA that can cause that to happen, that can make that occur, right? And there's a lot of specific damage that has to happen to a cell before it can become cancerous. But we will talk a lot about cancer, right? It is a very specific type of disease. But that being said, right, any kind of cancer is typically different than others. So it's really hard to cure cancer, although we have a lot of effective treatments for it. It's not something that can just be cured, right? If a cell becomes cancerous, we have to do something about it, right? And some cancers are worse than others. So you may have a lot of questions on those. I don't know everything about cancer, but we will talk about a lot of things. So just make sure that you keep those in mind. Ask me when we get to it later in class. Otherwise, you should be able to answer this question based off of skin cells. I just talked to you guys about that earlier. Right? Skin cells, you replace those every 2 weeks. So compared to other cells, would they have a long or short G1 stage? Explain. But otherwise, we need to jump to this cell size is limited. So it's all going to come down to surface area and volume. So there's a little bit of math that we need to do with this, right? And you have a table in your notes that I don't have. I have to recreate it up here. So I have these relative diameters of these cells. Now I'm going to assume that my cell is a cube and not spherical because it's just going to be a lot easier math. So I have a cube, right, that we know how to calculate surface area and volume for, right, because surface area would be just each one of those surfaces, length times width, times 6, because that would give me the 6 sides of a cube. Volume is length times width times height. So if I know the diameter of the cell, and I assume that it's 1 with generic units, I don't need to know what the units are, the surface area of that cell would be 1 times 1, which is 1, times 6 for the 6 faces. My surface area would be 6, right? So surface area for something with a diameter of 1 would be 6, whereas the volume would be length times width times height. 1 times 1 times 1 is just 1, so that should be pretty easy. And then I have a ratio, right, that in this case for surface area to volume is 6 to 1. So I'm just filling in that table. You should just do the same thing. So the next diameter, if it's 2, right, again, 2 times 2 is 4. That would be the area of one side. Take 4 times 6. That's going to give you your surface area. This time 4 times 6, that'd be about 24, right? Then I do 2 times 2 times 2 for a volume, and 2 cubed is going to be 8. So my new ratio is going to be 24 to 8, but I should be able to reduce that, right? So it's actually going to be 3 to 1 because 8 goes into 24 3 times. Then I increase my diameter one more time. If my diameter was 3, 3 times 3 is 9. 9 times 6 is going to give me, that would be 54, correct? And then my volume, 3 times 3 times 3. So 3 times 3 is 9. 9 times 3 is going to be 27, right? And 27 is half of 54. So this time my ratio is 2 to 1. So what you're supposed to be seeing here is that as I go across here, as my diameter, as my diameter gets bigger, my surface area to volume is actually getting smaller, right? So that's what I wrote right in here. As the cell gets bigger, that's my diameter, right? So as the cell gets bigger, my surface area to volume ratio is going to be getting smaller. Right, so that's going to matter for a couple reasons here. Right. Why do cells prefer to have a large surface area to volume ratio? It's actually a hard question, and it's a hard thing to understand, right, and to explain, but there's a couple different reasons. Right? First of which is that the surface area of the cell is representing that cell membrane. The volume is filled with that cytoplasm. You need certain things inside of your cells to survive. Right? Those things get into your cells through the cell membrane. If you can't get enough stuff into your cell, you can't survive. So the idea here is kind of a supply and demand. Right? And what you should understand from those calculations that we did is that your cell membrane is going to grow as the cell gets bigger, but your cytoplasm is also going to grow, and the surface area to volume keeps shrinking as that cell gets bigger and bigger because the volume grows faster than the surface area. Right? So as the cell gets bigger and bigger and bigger, the cytoplasm gets bigger and bigger and bigger, and so my demands in my cell get bigger and bigger and bigger. But the cell membrane doesn't get bigger fast enough to keep up with the cytoplasm. So as my demand in my cell grows, remember your demand is in your cytoplasm. You need stuff inside of your cells. That supply tries to keep up, but it can't, right? So my surface area and my volume are growing, both of them, but my demand is growing faster than the supply. So eventually you run into a problem. That's why there's a maximum in cell size, because at a certain point, the supply can't keep up with the demand. Right? It's kind of just a hard thing to walk your brain through. Right? So the demand grows faster than the supply. So eventually, the supply isn't big enough to meet the demand. Right? So that's one explanation, and it's just supply and demand. Right? If your demand is growing faster than your supply, eventually you're going to stall out. Right? Eventually that cell would not be able to keep up. The other idea is actually a little bit simpler, but it's harder to explain. It's easier to see visually. Right? Your cell has a size, right? And sometimes you need something to travel from A to B. And we talked about movement inside of a cell. And most of the time, you're completely dependent on diffusion to get things from point A to point B, right? So you're expecting molecules to move randomly, right? And just eventually make their way where they need to be, right? And sometimes they take weird paths, right? They don't always travel in a straight line, right? They're just moving randomly because their particles are in motion. They have energy because you're not at absolute zero. So these particles are just moving around randomly. Hopefully they get from A to B in a relatively quick fashion, right? But that's not always going to be the case. The bigger the cell, the farther A and B are apart from each other. If you're depending on diffusion to transport substances, you need that cell to be small, right? Because if A and B are close together, it's much more likely that substance can travel that distance quickly, right? So cell size is not just limited by the supply and demand, it's also limited in terms of transport, right? It's going to be hard to transport substances large distances when you depend on diffusion alone, right? So transport is going to be difficult, right? Because you need random motion to get things from point A to point B. So if A and B are close together in a small space, in a small cell, it's pretty easy to get things from one place to the other. But the bigger the cell, the longer and more convoluted the path that substance takes, the less likely it's going to make it, at least in time for that cell to survive and do the reactions it needs to do in a timely manner. You need that transport to be quick, not slow, right? So the smaller the cell, the faster the transport is going to be, right? Transport's my problem, but a small cell is going to be able to transport stuff faster because those things don't have to travel as far to get from point A to point B. It's fairly simple. So the supply and demand, right? Eventually that supply can't meet the demand because if the demand is bigger than the supply, the cell's going to be dead. So as soon as that demand gets bigger than the supply, which is happening fairly quickly, right, that cell's going to be in trouble. So again, we got surface area to volume. As the cell gets bigger, as the demand gets bigger, the supply gets smaller. So you need that supply to be big enough to meet your demand inside of the cell. You also need those distances to be short that need to be traveled because otherwise it's going to take forever. So again, you can answer this one. This first question with the help I've just provided, hopefully. Second question is a little bit easier, right? You just have to think about which thing's bigger and is it good to be big, right? Which one would have a larger surface area to volume ratio, right? So just remember that that is something that's inversely proportional. As something grows, right, the surface area to volume gets smaller. Should be a pretty easy question. Just get that answered on your own.